So the article I have chosen is resistance to freezing conditions of endemic Antarctic polychaetes is enhanced by cryoprotective proteins produced by their microbiome. And the microbiome plays a key role in the health of any organism. The host-associated microbiota can change from species to species based on environmental and biological factors. And one of the most important things the host organism can have is a stable microbial core. And nowhere is this more essential than in the waters surrounding Antarctica. Organisms living in Antarctica must adapt to the extremely cold conditions. Some do this by slowing embryonic development and growth. Others increase their oxygen consumption to maintain homeostasis in order to prevent liquid from freezing internally and to induce the antioxidant defense system. So in this study, the researchers investigated the microbiome of 3 polychaetes that were endemic and widespread in the waters of Antarctica. These polychaetes contain a stable bacterial core comprised mainly of Neothermus and Anoxibacillus. This allows the invertebrates to create this phylosymbiotic relationship with the bacteria. And the cryoprotective proteins that have been found in these invertebrates may be useful in developing some type of nature-based biotechnological application. So this slide shows the sampling area of the study, and it's going to show you the 3 different areas where they selected the polychaetes and also where they selected sediment. And this is a picture of the 3 polychaetes that they investigated in the study. I will not try to pronounce their names. I will just refer to them as L. gimminus, A. palmeri, and A. Drosophilus. 2 of these polychaetes are detritivores. So they basically feed on organic material, and those are going to be your L. gimminus and your A. palmeri. And the A. Drosophilus, which is the large one on the end, is actually a predator, and so they actually eat crustaceans and mollusks. So what the researchers also did, they did a taxonomic identification, as well as amplifying the select mitochondrial genes. So the taxonomic identification was done using what they call like a classical approach, which involved them using a dichotomous key while they were looking at the polychaetes under a microscope. And then the mitochondrial genes were sequenced, and the genes were 16S, 12S, and COI, which was your cytochrome C oxidase subunit 1. And so they did these things to basically ensure that they actually had the right organisms that they were wanting to study. And this once again just shows you the species that they selected, the areas that they collected the polychaetes from, the depth at which they collected the polychaetes, and the number of species from that group that they looked at. And of course, they also did an environmental sampling where they looked at temperature and salinity. And then they also did a relative abundance of the different sediments at the different depths. So there was gravel, there was medium coarse sand, there was fine sand, and then there was a silt clay. And this slide just also shows that they did a biochemical composition of sediments from the different sampling sites, and these are the 6 things that they actually measured from the sediment. Okay. So this graph right here, for this table, is showing the polychaete microbiome, and this was a taxonomic composition, and this is basically showing the relative abundance at the class level. And looking at it, you can clearly see that the 3 main class of organisms or class of bacteria is going to be your bacilli, your gamma proteobacteria, and your Deinococcus. But for now, we're not going to concern ourselves with the gamma proteobacteria. We're only going to look at the bacilli and the Deinococcus because that's relative to the Meothermus and the Anoxidacillus. So this is a second polychaete microbiome, and here is where they show the actual bacterial taxa at the genus level. And you can clearly see on this one that Meothermus, followed by the Anoxibacillus, is pretty well represented on here. Oh, and I should say, so for these microbiomes, they did a meta-barcoding analysis of the 16S ribosomal RNA genes of the 3 polychaetes, and it did yield 1,237 to 49,364 sequences. And that was after they did chimera removal and they did a denoising. And of course, like I said, the bacteria studied mainly belong to the Deinococcus, bacilli, and the gamma proteobacteria. And these 3 represented on average about 70% of the total taxonomic composition of the microbiome. But we're only going to be concerned with the Meothermus and the Anoxibacillus and not the gamma proteobacteria. So Meothermus and Anoxibacillus were shared among all of the specimens of the Algiminus and the A. palmeri, while Meothermus was present in actually all 3 of the polychaetes. Now, among the bacterial taxa, there were some that were exclusive to either Algiminus, A. palmeri, and A. Drosophilus. However, these exclusive taxa only actually accounted for less than 0.5%. So that so they weren't taken into consideration. Okay. So this is showing the taxonomic contribution of sediment microbiomes. And so the researchers did the sediment microbiome study in order to assess the origin of the polychaete microbiome. So the microbiome in the sediment was characterized by an operational taxonomic unit richness that ranged from 167 to 434. The taxonomic composition of the microbiomes showed no significant differences among the 3 areas. Now, there was similarity. The similarity among the samples was around 60%. They did have a dissimilarity, and that was mainly to the the different contributions of to the genus. One was Blastoparallula genus, which was anywhere from 1.7% to 18.5%, while the Flavobacteriaceae family was anywhere from 2.1% to 28.9%. Now, the microbiome of the polychaetes and the sediment were significantly different in terms of composition. And in this case, there was a dissimilarity of 97%. And that was based on the very low contribution of Meothermus and Anoxibacillus in the sediment. However, the main bacterial taxa of the sediment, which is the Blastoparallula and the Flavobacteriaceae, were actually present in only about 20% of the polychaetes that were tested. And so their contribution was less than 0.5% of that polychaete microbiome. Okay, so the results of the microbiome studies were confirmed by the construction of this operational taxonomic unit network, which highlighted the low number of OTUs that are actually shared between the polychaetes and the sediments, and the different links between the OTUs. Between the OTUs and samples are shown by these little gray lines. But it's pretty self-explanatory. Okay, so phylosymbiosis. So this picture represents the phylosymbiosis signals in the polychaete microbiome among all of the OTUs that were found. Only those that show a probability of phylosymbiosis up to 90% is shown. And the relative abundance is also shown for each polychaete specimen. And they did use an outgroup, which was another polychaete, which is the O. validus. And the phylosymbiosis analysis is carried out to investigate whether these microbial assemblages that were associated with these polychaetes had somehow maintained some ancient signal of host evolution. And they did find a congruence between the 16S mitochondrial DNA, which was based on this phylogenetic tree of the polychaetes, and also on the microbiome diversity tree. And they did detect a common ancestor between the two deposit feeders, which was the Albuminous and the A. palmeri. And there was another ancestor that was actually common to all three of the polychaetes. And this slide is basically just a little information about the Meothermus and Anoxibacillus. So Meothermus is a chemo-organoheterotrophic bacteria that can be found in both marine and terrestrial environments. And you'll typically find it at temperatures ranging from 40 degrees Celsius to 60 degrees Celsius. And of course, they have been found in subsurface polar habitats. And they can produce these thermostable polymers and enzymes that can degrade different organic substrates. And Anoxibacillus is mostly an anaerobic thermophilic heterotrophic bacteria. And they are able to degrade complex compounds. They have been found in the gut of non-polar marine organisms as well as in marine geothermal habitats. And they also can produce endospores. and other thermostable molecules. So this allows them to have a relatively high resistance to any of the adverse conditions they're in for an extended period of time. Okay, so this one is metabolic and functional traits of Neothermus and Anoxidobacillus. Okay, so the occurrence of Neothermus and Anoxidobacillus in the metagenomes of the annelid species available, the annelid species available in the gene banks were assessed through the 16S ribosomal DNA. And they did that by meta-barcoding meta-analysis, while the bacteria itself was investigated in a pan-genomic approach. So this meta-barcoding meta-analysis revealed that there was a lack of sequences that were related to Neothermus and Anoxidobacillus. So these lack of sequences were not found in any of the other annelid microbiomes. So the pan genome analysis of Neothermus was based on 16 genomes, and Anoxidobacillus had 43. And it revealed genes that coded for several of these functional traits that can be used for coping with extremely cold conditions. So there were these cold shock proteins and your ice binding proteins that they were encoded by the CSPLA and the IBPB genes in Neothermus, along with the CSPD, CSPB, and the IBPA genes in Anoxidobacillus. And these specific genes were identified in almost all of the genomes that they investigated. And then there was also genes related to acetyl coenzyme A metabolism and other energy generation, other ways to generate energy. There were compounds and enzymes involved in osmo and cryoprotection that were found in both the Neothermus pangenome with a coverage of around 87%, and also in the Anoxidobacillus pangenome with a coverage of 91%. And so basically, that's basically what this slide is showing you, is the pangenome coverage, everything that was found that helps them basically survive in this cold environment. And then on the left side, you can see all the numbers, the NCBI accession numbers in Neothermus genomes that were considered for the pangenomic analysis, and the same goes for the Anoxidobacillus genomes. Okay. So this one is a list of all these cold adapted proteins that were identified in the proteomes of the polychaetes. And so this is important because obviously the invertebrates themselves, they also had to produce some type of proteins to also help them survive. So it wasn't just relying on those two types of bacteria. They themselves also had these proteins that were also helping them to deal with these cold temperatures. So this, and the second one is also basically the same thing. Once again, it's showing these cold-related proteins that are actually specific to Neothermus or Anoxidobacillus. And it's also showing which of the host organisms that these bacteria are found in. So you have your host organism producing their own cold proteins, and then you also have help with the bacteria who are also producing their own. And this is just a picture. This is just the relative abundance of the bacterial cold-related proteins. And the polychaete proteins. So this, it was the Universal Protein Resource Code. Referred to the polychaetes constitutive proteins found here, and so it shows what it is for the 40S ribosomal protein, the 60S ribosomal protein, your catalase, and your histone 4. And the relative amounts of bacterial proteins compared to the constitutive ones of the polychaetes is expressed as a fold change. And it does show the little mathematical formula that you can use to get the fold change value. Okay, and this is just a slide, basically just explaining the molecular analysis of the microbiome of the polychaetes and the sediments. That they basically just extracted DNA from all these different tissue sites. So they did the oral cavity, the gut, the peripodes, and the tegments. They did this PCR amplification with a 400 base pair fragment of the 16S RNA gene with the hypervariable regions. And then they were purified and sequenced using the Illumina MySeq sequencer. And the sequencer used the same primers that were actually used for amplification. And then for the sediment, so that was for the polychaetes, the microbiome of the polychaetes, and then for the sediments, they did a total genomic DNA extraction. And then that DNA was pulled before they did the PCR amplification. And this is a continuation of the molecular analysis of the microbiome of the polychaetes and the sediments. And then this is just showing, like, for the Neothermus, basically they performed a real-time analysis of the 16SR DNA of Neothermus. And the primers that targeted the 109 base pair sequence were designed and synthesized by Eurofins. And then these primers were based on the 16 rDNA sequence that was actually detected during this meta-barcoding analysis. And so this picture right here, I like this picture because basically it basically just sums up everything. It's showing this common ancestor that these polychaetes had. It shows how negligible the Neothermus and Anoxia is in the sediment, but how prevalent it is in the polychaetes. So at some point in the past, the polychaetes and the bacteria did develop this symbiotic relationship with each other to help them survive. And they have these cryoproteins. Well, this is a good study example of learning more about cryoproteins and cryopreservation, because there are some industries, that rely on, like, tissue samples or reproductive organs or anything where they need to preserve tissue or any sample really for an extremely extended long period of time without damaging the sample. And so by looking at these cryoproteins that are being produced, there could be some type of, biological or biotechnological application that they can use from studying all these cryoproteins and enzymes that that the bacteria and these invertebrates use themselves in order to survive.